Now that we know a little bit about the structure of DNA and RNA, we can talk about DNA replication. Remember, the word replication means to copy. So basically, we're talking about the process of copying DNA. So why does DNA make copies of itself? It's to pass genetic information on to the new generations of cells. So the process of making new cells, if you think back, was mitosis. So it's the process of copying that genetic information to go through a cell that's going to go into mitosis. So replication facts. DNA has to be copied before a cell does mitosis or meiosis. DNA is copied during the S or synthesis phase of interphase, and new cells will need identical DNA strands. DNA replication occurs in the nucleus of eukaryotes. So remember, a eukaryote is a cell that has membrane-bound organelles, primarily a cell that has a nucleus. So the DNA replication is going to take place in the nucleus. So if we look at our cell cycle, we will see that interphase is made up of G1, S, and G2. This DNA replication is going to take place during the S phase of interphase, which happens before the cell does mitosis. I believe you need to write some of this into your notes. Please make sure that your diagram in your notes is labeled. DNA replication is said to be a semi-conservative model of replication. If we think about what semi means, it means partially, and to conserve something means to keep the same. So semi-conservative means to keep part of it the same, or two strands of the parental DNA separate and each acts as a template or model for a new complementary strand. And we'll look at this in just a minute. There's a lot of big words there. So over here you will see that I have my parental DNA. It's in blue. I have two blue strands. Those two strands are going to separate. Remember that the rungs of my DNA are made up of bases, and the bases are held together by weak hydrogen bonds. Those hydrogen bonds are going to break, and I'm going to separate it into two strands that are single strands. Those single strands then act as a template for the new DNA to form. Okay, so the two new cells in mitosis are going to have one strand from the parent and one new strand. This is why we call it semi-conservative. We conserve the parental DNA, but we do have some new DNA. Okay, so both cells end up with one strand from the parent and one new strand. The new DNA consists of one parental or original strand and one new strand of DNA, which is what I just explained. So DNA replication has some steps. The first step is that the enzyme DNA helicase unwinds and separates the two strands by breaking hydrogen bonds between bases. Remember that anything that ends in ase is an enzyme, and enzymes speed up reactions. So without DNA helicase, this process would take a very long time. The second step is free-floating nucleotides. Mainly you get them from your food, attached to each of the separate DNA strands. Then the enzyme DNA polymerase adds the nucleotides to start building the new strand of DNA. So if we take a look at this picture, you will see my DNA polymerase is adding my bases to my new strand.